Hello, everyone. I'm really happy that you're here with us today. And I'm going to start the session and going to talk with you about the B cell research solutions. I would say let's directly hop into the topic and start the webinar. So B cells have a lot of different functions in the immune system. So I just wanted to show you some to bring you all up to speed so that we're all starting from the same starting point. So B cells have many different functions like antibody production and memory function. So that's like, the main function that you know about B cells. But they also produce cytokines. They play a role in wound healing, in antigen presentation, also in the balance of Th1 and Th2 cells, dendritic cell regulation, also in tumor immunity and co-stimulation. So you see the B cells have many different functions that are really important in the immune system. In general, we'd always talk about the B cells, but there is no such thing as just like the B cells. So we have many different subsets, and even the subsets are distributed in different subclasses of B cells. So we have this follicular B cells. So these are the general B cells. So when we talk about B cells, these are these B cells, the B2 cells that we are talking about. It's the largest population of B cells, and it is a part of the adaptive immune system. But we also have the subclass of B cells that are called B1 cells. These are more part of the innate-like immune response. So they really are T cell independent. They have the same role as the normal B2 B cells, so they produce antibodies and play a role in the immune defense against mainly mucosal antigens. Um, but they're a bit different, so they have like a more, highly polyclonal BCR repertoire, and they can also bind to self antigens and mainly microbial antigens. And even this B1 cells have different subsets. There's like two main subsets, the B1A cells and the B1B cells. So you see, this is already starting to be like, very confusing, and we have like so many subsets, even like in this two different types of B cells. So that's why obviously it's good that we talk about the B cells today. The B cell development starts actually in the bone marrow. There rearrangement is happening to really get the functional B cell receptor. Then, of course, positive and negative selection of the B cell is happening that are antigen independent, because like in the bone marrow, we don't really have the antigens where the B cells can bind to. But these processes are really important, especially the negative selection for the central tolerance. When we then have the mature B cells, they develop into follicular and marginal zone B cells. And the follicular B cells are mainly in the blood, so they get activated when they get contact to an antigen. And when they are getting activated, they normally undergo this German center reaction. But some of them are directly going into be differentiated into short-lived plasma cells that then produce antibodies. But as the name say, they're only short-lived, so they don't survive for long. But the normal way of development is that they actually undergo this germinal center reaction where a lot of different processes are happening. They do proliferation, maturation, also apoptosis is happening there. And then, of course, like the stimulation and the interaction with the T cell. So this process is like really important to get really highly specific antibodies. And when the B cells are then done with this germinal center reaction, they can then be differentiated into memory B cells to build a long time memory. And then we have like this long-lived plasma cells that survive for much longer and produce antibodies over a lot of time. And also we have direct development into plasmablasts and then into short-lived plasma cells to quickly produce antibodies against pathogens. But then we also have the marginal B cells that are not undergoing this germinal center reaction. So when they get the antigen contact, they are also getting activated and build a plasmablasts and then plasma cells. And this is like mainly a T cell independent plasma cell production. So you see there are many different ways and many different subsets of B cells. But of course, I shouldn't forget that we also have regulatory B cells that is still like in a discussion, like what is like, the way of development for this. But so far it looks like that actually probably any type of B cell can be differentiated into regulatory B cell subset depending on the stimuli that the B cell is getting. And when we're talking about B cell, an important workflow is obviously the therapeutic antibody production. So this is a workflow where a lot of people are working with. So you can start with human or mouse materials, depending on what are you doing. If you maybe use like naturally exposed human, that was like done a lot for COVID studies, but also a lot of mice are getting immunized for several diseases, and then people want to isolate the B cells to get antibodies against those antigens. And the first step of this workflow would be then the B cell separation so that you get like enriched B cells. And then obviously, as you're working with an antigen, you want to have a specific antibody against your target. You can go into sorting of antigen-specific B cells. And then the next step would be the B cell cultivation because usually these antigen specific B cells are not too many, and to be able to further analyze the antibodies, the binding, the specificity, and also be able to maybe like, clone or sequence those cells, it is nice to expand them in culture. Then the next step, you can do the binder selection so that you really know what antibodies are binding nicely to your target and if they're binding great to your target. That is really important. And then you get your lead candidates of antibodies that you can further analyze. And of course, here functional testing is really important where you can do a lot of assays to analyze such antibodies. Of course, this can also be done with materials like splenocytes, lungs, or maybe even tumor materials. And there, of course, you would need first a step to do like some sample preparation to prepare your cells so that you have a single cell suspension to go into B cell separation. So this is obviously also very important. And now I would like to discuss with you some challenges that you might have in these different steps of the workflow and give you some hints how you can find a solution for that. So usually it's like quite complicated, as I showed you in the beginning, that we have many different subsets, so many different kits are available to isolate the B cells. The B cells are really sensitive, so it's like not so easy to have like really viable B cell for all subsequent steps. And also the RNA quality is really important for single cell sequencing, but also for cDNA synthesis. To show you what is like, possible is like, from human, you can have many different starting materials as you see here. And for mouse, of course, we also have a lot of solutions for you from all the main important starting materials where you can work from mice. And as I said, sometimes it might be really confusing to find the right solution. We collected all the different materials and all the different subsets and kits of interest for you. So we have a nice B cell brochure and a nice poster where you can look into, okay, I want to have this specific subset of B cells, and here is like the starting material. Okay, I need this and this product, this and this separation product to actually get my B cells. So here you will get all the information nicely collected for you so that you know what is of interest for you. And also we have some nice application pages for the B cells where you can look into all the different workflows we have for the B cells to get like more information on, yeah, example data, what like, what is to expect, what are steps, where can I find some help. So here you can also then later when you get the slides, click on this picture and then you can find directly the information on the B cell application workflows. And I just wanted to show you some example data. It's also what you can find on our web pages and on all our materials, that this B cell isolation is really gentle. So we tested the positive and the negative separation of the B cells here with the pan B cell isolation kit, and also here with the Release CD19 microbead kit. And you can really nicely see that the B cells after separation, no matter what technique you use, are really not activated. So we really tested for all the activation markers, CD86 and CD80, that are really important. And you see all of them look the same. So no matter if you're having untouched isolation or positive isolation. Then we cultured those cells for 7 days and you see still the cells are not stimulated. So all the cells, or almost all the cells, are double negative. And only when you add stimuli like CD40 ligand and IL-4, the B cells are getting activated. So you see it's no activation of the cells, no matter what technique you're using. Also, we collected some data from the isolation of B cells from leukopak and check the RNA quality of this purified B cells. And we could nicely show that the RNA quality of the B cells is really high. So when you look here, the RIN value of the RNA, so 10 is the highest, so the best quality is really great and really high. So you can nicely do any sequencing, any cDNA synthesis when you want to use like the RNA of the B cells. So then the next step of the workflow would be the sorting of the antigen-specific B cells. And this is like, really tricky because these cells are really rare. They're very sensitive. We already talked about this. And of course, like, sample carryover or contamination, this could be a problem. And of course, when you're working with infectious materials, the high safety level for sorting is sometimes a problem. And we actually really have a very nice solution for you, the gentle sorting with the MACSQuant Tyto, because it's gentle and fast. It's a very easy system. It is an operator-free sorting and actually operator safe because you have the closed cartridges. It is high cell viability, low pressure. That would also lead to the high viability and is very sensitive to your cell, and you don't need any cleaning. And here we have an example of B cells that we sorted on that material. So it is like, here spike positive B cells that we sorted. And you see in the starting material, we only have 0.35% of antigen positive B cells. So it's just a few. And when you really look then how the sorting worked, it is really nice. We were able to gain 91% purity for our really rare B cells that are actually IgG positive spike B cells. So it's not only spike positive, so we could even look into the IgG positive spike B cells and see that they are expressing, like, you see here down here, they are expressing like all IgG. There's just some that are expressing IgA. And the viability of these cells were actually 98%. So it's really great technique or instrument to sort antigen-specific B cells. And then also we have the characterization of the B cells is really important. So there, the REAfinity antibodies are helping a lot. And I collected a nice panel that we usually use for B cell phenotyping, where you can look into all main B cell subsets that are usually looked into. So you can look in for the B cells, then memory B cells, if they are switched with IgD, Then for plasma cells, we have two markers in, and then also for the activation, we have two markers, CD86 and MHC2. And with the use of the REAfinity antibody, this is like a really highly valuable panel. And if you want to know more about this, please later when you have the slides, click on this button and you can watch the webinar where we talked in detail about the B cell analysis. Now I will just like show you some challenges and solutions for the B cell cultivation. So this is sometimes a bit tricky because it's not so easy to cultivate B cells. As I said, they're really sensitive. It's sometimes not working nicely. Cells are not growing. It's not nice expansion and inconsistent. And it's really time consuming actually to set up such a protocol that it is working. Why is this so complicated? Because in vivo, the B cells are getting several signals to be activated. So first they bind the antigen, what is like, the first signal of activation they're getting. Then usually the T cells are coming, presenting via, binding via the T cell receptor to the B cell that is presenting the peptide on MHC2. And then also the CD40 ligand is binding to the CD40 receptor on the B cell where the coll stimulation is happening. And then, of course, the T cell is producing the mixture of cytokines that is then leading to the specific differentiation into the needed B cell subset. So all the stimuli are happening to get a nice clonal expansion and differentiation of the B cell. So this is really hard to mimic in vivo, and that's why we actually produced this nice kit. It is called the B cell expansion kit human, where we have all the ingredients in that try to mimic this stimulation. So we have the CD40 ligand with a cross-linking antibody that is nicely mimicking the activation of the T cells. And then, of course, we have the cytokine IL-4 that is also giving the stimulation via the cytokine receptors. And by this, you get a nice expanded and activation of B cell. So if you want to know more about this, we also have a webinar on that. So please later click here to see the webinar on B cell culture. So let me summarize now what solutions we have for you for this or any B cell workflow. So we have a lot of different kits that you can use to isolate your B cell subset of interest, no matter which starting material you have, if you have blood products or if you're starting from PBMCs. We have different kits, and as I said, just check out our homepage or our B-Cell Quick Guide to find the product that is most suitable for you. Then we have the MACSQuant Tyto that's nicely sorting antigen-specific B cells and high viability and purity. And then we have the B cell expansion kit that helps you nicely to culture your B cells of interest. Of course, we also have the CD40 ligand multimer kit, TLR ligands, and other cytokines that help you with the B cell culture. And then, of course, antibodies and analyzers to analyze your B cells of interest and the antibodies. For the mouse workflow, we have, of course, if you're starting from spleen or any other tissues, dissociation kit and the gentle max that is helping you to get a nice single cell suspension. And of course, we have also the specific mouse kits that are helping you to isolate your mouse B cells. Of course, for mouse, you can also use the MACSQuant Tyto and the Max antibodies to sort your antigen-specific cells. For mouse, we also have TLR ligands and MAX cytokines for the B cell cultivation. And of course, for the selection of your binder, the selection of your B cells, we have the MAX antibody and the MACSQuant X that is helping you a lot with this workflow. Here are also some contacts that you can use. So if you have questions, no matter in which country you are, we have a support for you, a local organization that you can contact and write an email to if you have any questions and if you need any help. And of course, always remember to visit our homepage to get some more information on the topics, or maybe see some more of our nice webinars that are giving you a nice introduction on all our solutions. And then I will thank you for your attention, and that was it from my side.